Students, once we are familiar with all the concepts that we have in inflammation, now let's go on to the core inflammation that not only is considered difficult, but is also a very high yield potential topic that we have in the main exams that we appear for. What is that topic? Those are referred to as the changes in inflammation. The changes in inflammation. Now students, remember, whenever we are talking about changes, we are going to focus on two things. The first one is going to be the vascular changes. What are the changes that are taking place in the blood vessels? Second are going to be the cellular changes. Now remember, when you're talking about vascular changes, which vessels is the next question that should come to your mind? Everyone knows that arteries further divide into arterioles, which further branch into capillaries. They combine or they receive the blood into venules, finally into veins. This is a basic concept that everyone has. Out of these, starting from arteries and ending till veins, which is the blood vessel which is going to show changes of inflammation or vascular changes? That is going to be the arterioles, the capillaries, and the venules. Arterioles, capillaries, and venules. So you can say cut out the two ends, cut out the arteries, cut out the vein. What we have in between, arterioles, capillaries, and venules, those are the ones which are going to show changes of inflammation. What about the next category when we say cellular changes? What are the cells that we are talking about? Primarily, remember, these are the white blood cells. Leukocytes, as you also can call them. White blood cells or the leukocytes. Now, these are very important two changes that we have to deal with, vascular and cellular. And in the same order, we are going to begin. First, we are going to read vascular changes. So what is the trick of reading these? Let's make a list. Let's know the names first and one or two important questions on each. Then we are going to go into a detail. So first, let's begin, students, with vascular changes. So what is that list that you need to know in vascular changes? The first change that we have over here is vasoconstriction. Now please keep this in mind. This is one of the questions which is incorrectly answered by many. Vasoconstriction. The vessel diameter constricting, becoming smaller. Vasoconstriction. The second vascular change that we have is vasodilation. The vessel is going to dilate. The diameter of the vessel is going to increase. The third vascular change that we have is increase in vascular permeability. Increased vascular permeability. And the fourth and the last one that we have is stasis. You don't have to mug up these, you don't have to mug this list up. For the time being, what you need to know, this is the order in which we will be studying this topic. So first question, if I put up to you over here, which is the first vascular event to occur? From this list, you can easily guess the first vascular event that we will have. The first vascular event of inflammation will be vasoconstriction. Now, if instead of mentioning vascular or cellular, if the question just says first event of inflammation, then also your answer will be vasoconstriction. So, vasoconstriction is your answer for first vascular event also and first overall event also. One question on this list is complete. The second question that you are asked, a very favorite with the examiners, if someone asks you, out of the entire acute inflammation that we will study, what is the hallmark of acute inflammation? Now, what of these will be the hallmark? So remember, increased vascular permeability is the hallmark of acute inflammation. This is the hallmark of acute inflammation. Very important. We have multiple hallmarks as we go through general pathology. We started that. If you can recall from cell injury, one hallmark of Barrett's esophagus was the formation of intestinal columnar epithelium. We've added on to that list by discussing apoptosis also. What was the hallmark of apoptosis? Correct, it was nuclear chromatin condensation. What was the hallmark of neuronal apoptosis? Neurons, if you can recall, did not have caspases. So over there, it was activation of the apoptosis inducing factor. Those were few hallmarks that we did. We've added on to the list. What is the hallmark of acute inflammation? It is increased vascular permeability. Now students, with this particular concept, let's take up each one of these steps in detail. The first vascular event that we are going to read, students, and I hope everyone can recall, the first vascular event was vasoconstriction. Now, as everyone knows, vaso means vessel. Constriction means that the diameter of the vessel is going to become narrow. Constricting. What are the questions that you need to know over here? Remember that this is the first event, and by now everyone knows this. Second point is that this is going to be an extremely transient phenomena. Now students, this will last only a few seconds. But although it lasts only a few seconds, it still is the first event to occur. So the first event, transient phenomena, and the next question, why is it happening? Remember, this is nothing but a reflex phenomena. This is a reflex phenomena. So these 3 points about vasoconstriction: first event, transient, only for a few seconds, and reflex phenomena. These are important to know. Next, what was the second event that we need to deal with? That is vasodilation. Again, focus on the term. Vaso means vessel. Dilation means that the diameter is going to increase. What do we mean by this? So, for example, if this is a normal blood vessel, but in inflammation, what is happening? The diameter of that vessel is increasing. So what is happening? Vasodilation. Now what will happen when the diameter of the vessel increases? If this was the normal blood flow, on dilation, the blood flow to that particular organ or area or tissue is going to increase. Once the vessel dilates, the diameter increases, the blood flow is going to increase. Now students, just by applying a little bit of common sense, if the blood supply to an organ or a tissue increases, what will happen to the color of that area? Definitely it will become reddened. So there is redness. Please recall, which particular cardinal sign would you call this? Remember, R for redness and R for rubor. Second, if the blood supply to a tissue or an organ increases, what will happen to the local temperature of that organ? There is going to be, correct, an increase in temperature. Which cardinal sign are we talking about now? Remember, increase in temperature was calor. So 2 cardinal signs we've done. We've done rubor, that is redness. We've done temperature increase, that is calor. All of this is happening because of vasodilation. Now remember that why is vasodilation happening? What is the mediator for this vasodilation? What is the mediator? And remember, this is one of our favorite mediators in acute inflammation, that is histamine. So vasodilation is because of histamine. Remember, on the contrary, vasoconstriction, which was the first event that we had, that was actually a reflex phenomena. Vasodilation is because of histamine. Now the vessel first constricted. After that, for a very few seconds, Few seconds after that, it dilated, that is vasodilation. Now we move on to the third event of inflammation, vascular event, that is increased vascular permeability. Increased vascular permeability. Now students, remember, for this you've already done one question, one very important MCQ. This happens to be the hallmark of acute inflammation. This is the hallmark of acute inflammation. What next? If you have to understand this particular concept, so let's take up a blood vessel. For example, if this is a blood vessel, we just discussed that what is the lining of a blood vessel? The lining of a blood vessel are these cells, and what do you call these cells? These are the endothelial cells. These are the endothelial cells. Now, one question about endothelial cells, students, I want to finish here. If someone asks you the marker of an endothelial cell, remember it will be CD34. Very important question, has been asked earlier. One of the collateral questions that we need to know. Marker of endothelial cell is CD34. Now coming back, what do you mean by increased vascular permeability? Means the permeability of the vessel is going to increase. How? Some changes are going to happen in these endothelial cells, because of which all of what is flowing inside the blood vessel is going to come out. Students, what is flowing inside the blood vessel? We have the plasma. So the plasma will come out. Everyone knows that the plasma has proteins. So the plasma proteins will come out. Plasma proteins will come out. Also what we have are the leukocytes or the cells. So the plasma proteins and cells will come out once the permeability increases. Whatever was flowing inside comes outside. These three together are famously called as exudate. Famously referred to as exudate. Now, fluid and cells from the vessel has come out into the tissue. In the tissue, all of this starts accumulating. The plasma, the proteins, the cells, they start accumulating. What will they form over there? An accumulation, or you can say a swelling. So there is going to be a swelling. Again recall, swelling was which cardinal sign? It was tumor. It was tumor. So you've now done the reason for tumor or swelling. That is because of increased vascular permeability. Very important. Now there is one more MCQ that is a favorite with the examiners. We just discussed that vascular events will occur in arterioles, in capillaries and in venules. But if they ask you out of these three also, which is the vessel which is maximally going to show? So maximum increased vascular permeability is seen in which vessel? Remember, the answer to this will be venules. Venules are going to show maximum increase in the vascular permeability. So regarding increased vascular permeability, you've done two questions. Number one, hallmark of acute inflammation. Number two, maximum changes are seen in venules. Now students, just by saying that there was a blood vessel and the permeability increased and swelling occurred, it is not as simple as it just sounded. There are certain mechanisms by which this vascular permeability is going to increase. There are certain mechanisms, a number of mechanisms actually, that you need to know. Now because there is a long list of these mechanisms and there are certain points of differentiating each mechanism from the other, this tends to get a little confusing, and whatever tends to get a little confusing for you will definitely be a very favorite topic with the examiners for the exam. So what we are going to do, we are going to figure out a trick or a strategy of how to simplify these mechanisms and make a certain concept which will last us a longer time in the exam. So let's talk about the first mechanism. Mechanism of increased vascular permeability. Now you know that lining of blood vessel is endothelial cell. So for example, if this is the blood vessel and these are the endothelial cells. How will something that is flowing inside come outside? There has to be some gap or some area, some way or path that has been created for it. How will a path be created over here? If the two endothelial cells contract, if they contract, the gap between them will increase, correct? So the name of the mechanism is endothelial gaps. Or how are these endothelial gaps happening? Because the two endothelial cells have contracted. If they contract, the gap increases. So I am going to call it endothelial cell contraction. Endothelial cell contraction. Correct? So whatever was flowing inside will now come outside because there is a gap. Now there are one or two important questions that you need to do over here. Question number 1. Out of a number of mechanisms that we study right now, remember this is the most common mechanism of increased vascular permeability. So we'll do a long list, but out of all of those, this is the most common that we have. Question number 2. What is going to mediate this? What is the mediator for this endothelial cell contraction? Under the influence of what will the endothelial cells contract? So our favorite mediator is histamine. So this is primarily under the effect of histamine. Very important. Also can be attributed to bradykinin. So these are mediators which we will be discussing in inflammation in great detail because these are extremely important mediators. But for the timing, you need to know that this is because of histamine and bradykinin. What next? The question number 3, which I was saying makes every mechanism confusing from the other one, is the timing, or you can say the duration. What do I mean? For every mechanism, they will ask you, is this mechanism going to happen or occur quickly, or is it going to take time before it occurs? Will it finish off very early, or will it be prolonged? So the timing of every mechanism is very important. Now remember, endothelial cell contraction is because of which mediator? Histamine. As we will read later on, histamine is a preformed mediator. Histamine is a preformed mediator, means it is always present in our body. All that has to happen now is when inflammation starts, it has to get released, right? So will the body take any time? No. This mediator is already present. Inflammation starts, within seconds it is released. So will this response be immediate or is it going to be delayed? Now you can clearly understand that this particular response is an immediate response. Again, remember histamine is not something that is very long acting. Histamine is short acting. So for how long will this mechanism occur? Again, for a shorter duration. So I can say it begins very quickly and it is going to be a very short-lived response. So I am calling it an immediate transient response. Students, a very important question that is asked, the timing of these mechanisms. So if you know the underlying concept, the timing will be easier to remember. So now you know that endothelial cell contraction is by histamine. Histamine is preformed, so the timing will be immediate and transient. Now students, there is one mechanism more which is not dealt with in many of the books in detail, but just by a change in the term, the entire answer to the question is going to change. The first mechanism was endothelial cell contraction, the most common. The second mechanism that we are going to have is again to do with endothelial cells, that is endothelial cell. But now instead of contraction, we are going to use the term retraction. We are going to use the term retraction. So there is only one term that has changed. Again, the same thing is going to happen. If this is a blood vessel and these are the endothelial cells, the same thing is happening. Whatever was flowing inside will come outside. Why? Because there is some path that is created. But over here, the underlying mechanism is different. Instead of a contraction, the endothelial cells have retracted. Sounds similar, contraction, retraction. You would say that ultimately, if you talk in simple English, it sounds quite similar, but the underlying mechanism is different. Contraction was because of histamine. Retraction, on the other hand, is because of cytoskeletal reorganization. It is due to cytoskeletal reorganization. Now students, please remember, the cytoskeleton of every cell in our body has a certain framework. It has a cytoskeleton. If you want to change the cytoskeleton, or if you want to change the framework of a particular cell, it is not going to happen within seconds. It is going to take time, right? Also, once it is changed, to come back to the normal process, once inflammation is over, for this cell to come back to its normal, again the framework has to change to back to normal. That is also going to take time. So will this be an immediate transient response? No. Because this is going to happen because of changes in the framework, remember that the timing or the duration of this particular is delayed and prolonged. This is a delayed, prolonged response. So students, endothelial cell contraction was because of histamine. Preformed mediator, quick response, immediate and transient. Endothelial cell retraction is because of cytoskeletal reorganization, will take time to change the framework. So delayed, prolonged response. The next question that you will get, what is the mediator for this change? Why is the framework of the endothelial cell and how is it changing? It is because of the mediator that we will also read eventually is cytokines. What was contraction because of? Histamine. What is retraction because of? Change in framework because of cytokines. So we've done two very important and the most confusing mechanisms that we have. The third mechanism that we have is referred to as endothelial injury. Endothelial injury. Again, let's take the example if this is a blood vessel with the lining cells. Endothelial injury means that there is something that is coming and injuring the lining or the endothelial cells. Is it necessary that it will only injure at the junction of the two endothelial cells? No. Injury can happen anywhere. So now endothelial cell contraction and retraction was that the two cells are moving apart and at the junction the permeability increases. But in endothelial cell injury, anywhere along the endothelial cell the injury can happen. So anywhere the permeability can increase. It can be at the junction, it can be in the cell, anywhere. Very important, this is where the examiners play around with the language the most. They will give you a scenario and you will have to categorize that the injury that they are talking about, is it a mild injury or is it a severe injury? So remember, the examiner can either talk about a mild injury, or the examiner will talk about a severe injury. And how will you know that? By certain examples you will be able to easily differentiate. For example, the examiner says that the person suffered from a delayed sunburn. Delayed sunburn. What do we mean by this? Now, sunburn is a mild injury. All of us have experienced it or have seen others who have been through this. Does a sunburn start immediately as you enter the sun? No. On gradual exposure to higher degrees, soaring temperatures, the sunburn sets in gradually. And if a person has a sunburn, will he recover within seconds or within minutes? No. Again, the recovery also takes time. So what would be the timing of such a response? It will set in gradually, it will recover gradually. So I will say that the timing of this particular response is a delayed, prolonged response. Delayed, prolonged response. Now students, please, as we are proceeding, try to recall. Endothelial cell contraction was immediate and transient. Endothelial cell retraction was changes in the framework. It was delayed and prolonged. Injury, when we are talking about a mild injury like a sunburn, it will be delayed and prolonged. But if we are talking about a severe injury, what severe injuries do you need to know of? Like we have burns. In burns patient, the injury is definitely severe. If someone has severe infection or sepsis, so severe infections. If there are some toxins which are present in the blood or some poisons which are present in the blood, they are definitely severe. Poisons and toxins are severe, so they will damage the endothelial cells. So all these come under examples of severe injury. Now, for example, a toxin or a poison or burn, the best example can be burn. You know that once a person suffers from burn, the change happens immediately. The damage happens immediately. So what type of a response would it be? Immediate to start with. For the recovery of a burns patient, you all know it takes time. It takes many weeks and months for a burn patient to recover. So now what would be the timing of this response? Severe injury will begin immediately. For example, in burn, the injury will begin immediately but will take its own time to recover. So this is an immediate, sustained response. This will be an immediate, sustained response. Right? So we've done, please read the question. When the examiner is talking about endothelial injury, he will definitely categorize either it into a mild injury, which will be delayed and prolonged, or a severe injury like burns, which will be immediate and sustained. So we've done 3 mechanisms of endothelial cells leading to increased vascular permeability. The fourth mechanism that we have is known as a simple one from the name itself, leukocyte-mediated response. Means that, for example, if this is a blood vessel and these are the endothelial cells. These are the endothelial cells. Leukocytes, for example, I hope everyone can now recollect which particular leukocyte am I drawing with these 3 lobes. Correct, it is a neutrophil. So the neutrophil or any leukocyte is going to come and is going to damage these endothelial cells. So what are we calling it? Leukocyte mediated. Why? Because these leukocytes or white blood cells are going to release certain enzymes. So the leukocytes come and they release enzymes, and these enzymes then kill the endothelial cells. A simple mechanism to remember just from the name, leukocyte-mediated response. The fifth one that we have, students, is a more newer one that you need to deal with. That is something known as transcytosis. Now, again, our favorite concept is that we need to break the term, and if we break the term, we'll get the meaning. Trans means through something. Through what? Cyto means cell. So over here, the vascular permeability is going to increase because there is passage through the cell. Let's understand this with the help of a diagrammatic representation. For example, if this is the blood vessel that we have, lined by these endothelial cells. Now, through the cell means somewhere along here the movement is going to occur, through and through the cell. Now, cell is a barrier. How can something just pass through the cell like this? Means something over here has formed. Some channel over here has formed which is allowing the cells to move out. And this, students, is known as the VV organelle. This is the VV organelle, also known as the vesicular vacuolar organelle. Vesiculo-vacuolar organelle. So, through the cell, permeability will increase when this VV organelle is formed. And how is this VV organelle formed? What are the mediators? There are two important mediators that you need to remember. These include our favorite mediator, that is histamine. And we have, just by the name you will be able to memorize, something is forming in the endothelial cell. Some organelle or some pore or channel is forming in the endothelial cell. So the name of the factor should also have endothelium in it. That is vascular endothelial growth factor. Vascular endothelial growth factor. So these two mediators will result in the formation of vesiculovacuolar organelle and the vascular permeability will increase. So students, this entire thing is known as transcytosis. The last mechanism finally that we come to in this, the last mechanism that we have is something known as angiogenesis. Again, something that you already have heard of multiple times. Angio means blood vessel, genesis means formation. So this is known as the formation of new blood vessels. Formation of new blood vessels. Now students, one concept that you need to understand right now. For example, I am taking the cross section of a blood vessel, a new blood vessel, a cross section. And lining will remain the same, that is the endothelial cells. A new blood vessel will also have endothelial cells, but can you notice one striking difference in this diagram versus the other diagrams that we had drawn? Do you see gaps present over here? When a blood vessel is newly synthesized, when the blood vessel is new or anything in our body is new, that is going to be immature. Whenever a new cell, a new structure is formed in our body, it is going to be immature, and with time it is going to then mature. What is the characteristic of an immature blood vessel? And an immature blood vessel is also known as a leaky channel. Is also known as a leaky channel. Means that the endothelial cells have not fused with each other. There are these gaps present. And if gaps are present, what will happen to the plasma and the cells flowing inside? They will come out. Indirectly, what are we talking about? Increased vascular permeability. So remember, angiogenesis is a mechanism of increased vascular permeability. Why? Because the new or the immature blood vessels are leaky channels. Why are they leaky? Because the endothelial cells have gaps present between them. Over a period of time, these gaps are going to fill up and the vessel becomes mature. Now remember students, for everything we were doing mediators. So what are the mediators that we have for angiogenesis? These are the same mediators that we had for transcytosis. Please recall, those were our favorite, that is histamine and vascular endothelial growth factor. Again, we are growing new blood vessels, so we need a vascular endothelial growth factor. So these were the 6 mechanisms that you had to do. Out of these, please remember, the most common mechanism of increased vascular permeability was endothelial cell gap because of endothelial cell contraction, which was mediated by, correct, histamine. And histamine is a preformed mediator, so the response was immediate and transient. Now I want you to go back to the fact that we were discussing vascular events. The first vascular event was vasoconstriction. The second vascular event was vasodilation. The third one, which is actually the hallmark, that is increased vascular permeability. And the fourth and the last one, probably the shortest as well, was, please recall from your list, it was stasis. It was stasis. Very simple to visualize now that, for example, if this was a blood vessel with, yes, the endothelial cells, the lining, why will stasis happen? Because everything that was present inside the blood vessel has moved out. Everything has moved out, like the plasma, the proteins, the various types of cells have moved out. What are we left behind inside the cell? When the WBCs have moved out, what has happened to the concentration of the RBCs inside the blood vessel? Definitely, if all the plasma, if all the fluid has moved out, the fluid inside is less. If the fluid inside is less, this means that there is an increase in the red blood cell concentration. There is an increase in the red blood cell concentration, right? If the red blood cell concentration increases, indirectly what is happening to the viscosity? There is an increase in the viscosity of the blood. And students, what is very evident is that once the viscosity of the blood increases, the blood flow is going to slow down. So there is going to be sluggish blood flow. And what is sluggish blood flow known as? Exactly our fourth mechanism or fourth event, that is stasis. So because the RBC viscosity and the concentration increased, the blood flow decreases and the fourth vascular event is complete, that is stasis. So we had 4 vascular events to recall. The first one was vasoconstriction, which is a reflex phenomena. The second one is vasodilation because of histamine. The third one was the hallmark, that is increased vascular permeability. This was happening where the most common mechanism, that is endothelial cell contraction, which in turn was mediated by, again, histamine. And lastly, after permeability increases and all the fluid moves out, we enter into the fourth vascular event, that is stasis. So this was the entire summary of the vascular events.